Okay, it's concept 9.4. We're going to finish up much of the chapter today and we'll have only learning it all left. We'll start on oxidative phosphorylation. If you remember, oxidative phosphorylation is where we're going to get most of our ATP. So surely this is a very exciting day. Now I want to remind you what the video we watched on, it wasn't Netflix, BioFlix told us. We generate something like 10 million ATPs per second per muscle cell. Now that is an awesome number, and I assume that's on an average second, not while we're playing soccer, etc. So anyway, oxidative phosphorylation is where we're going to generate most of that ATP. Now, following glycolysis and citric acid cycles, we have ferried our NADHs and our FADH2s, our electron carriers, off to oxidative phosphorylation. Now, before we even begin, I think I'll show you this electron transport video. I have another one I may show at the end because this is by far the most complex part of the cycles.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD plus and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay. The electron transport chain, all of those complex multi-proton complexes, it's protein complexes, are found in the cristae of the mitochondria. Now think about the shape of the cristae. It's kind of an infolding of the inner lining or the inner membrane. Notice that folding pattern is going to give us extra surface area for more electron transport chains. That was designed by a mighty designer. The carriers will alternate. They'll drop their electrons off. They'll also drop off protons, which are the hydrogen ions or protons, and they will be, the electron carriers will be oxidized by the proteins in the electron transport chain. The proteins in the electron transport chain will be more highly electronegative than the electron carriers. So they'll steal away the electrons from the carriers, and each protein in the chain going down to the bottom of the electron transport chain is more highly electronegative. Thus we have the flowing from the first protein in the electron transport chain to the last one. Now each time the electrons move from one protein to another, they're going to lose free energy, but the energy they lose will be made into the ATP. At the very bottom of the electron transport chain, you will get oxygen, which is a very potent oxidizing agent. it will be reduced with the electrons. Hydrogen ions will also be sitting there, and water will be made as a waste product. So let's look at this. This picture is in your book, and again, I recommend you really study pictures. This is the first protein complex, and we've got 4 of them in the electron transport chain. These are called cytochromes. Most of them are organic molecules. You do not have to know the names of any of them, but the electron carriers will drop off their payload of electrons, and this cytochrome here is more highly electronegative than the NADH. So therefore it steals away the electrons from the NADH, but what it will also release is the hydrogens. These hydrogens now will flow through the electron transport chain. So as the electrons are flowing down the electron transport chain, hydrogens will be pulled to the intermembrane space, and they will create what we call a proton motive force. We'll see another diagram of that in a minute. But for now, each of these proteins are more highly electronegative than the last, and so each one will start out being reduced, and then it will be oxidized as its electron changes places to this one. This will be reduced, and then it will be oxidized when it loses its electron to this one, all the way down to the bottom where the ultimate electron agent will be the oxygen. It is the most potent of oxidizing agents. Okay, one more thing. Here's our FADH2. Notice that NADH drops its payload at this very first cytochrome in this first multi-proton complex. FADH starts here. Therefore, it skips some of this energy harvesting right here. So in so much as it starts the electron transport chain a little late, it will not be able to, shall we say, accomplish as much work as NADH. Now I want to look at this picture. You can go back and read the words to the last slide tonight if you want to download the PowerPoint off my website, but I'm going to explain all of that as we look at this very, very excellent picture. Be sure to know this picture for your next quiz or test. This is oxidative phosphorylation, and it is made up of 2 subcycles, shall we say. Oxidative phosphorylation starts with the electron transport chain. It will create a proton motive force, and then it will go through chemiosmosis. So notice these sub stages down here. Here's NADH. It drops off its electrons, which float through the electron transport chain. Each of the proteins will be more highly electronegative, pulling the electrons down to the final electron acceptor will be oxygen, and that would be happening down here. Now, not only does NADH drop off its electrons, it also drops off hydrogen. So notice this has lost its hydrogen. The hydrogens are pulled through these protein complexes, and it will create a very high concentration gradient of these hydrogen ions. Well, you notice that a hydrogen ion is creating acidity, but it's also a proton, so we're going to call it a proton motive force. Now remember, whenever we have a high concentration gradient, we have potential energy. As the concentration gradient of these hydrogens get more and more concentrated, they will, as it were, be popping the seams to escape. Well, the only place that they can escape is this ATP synthase. So these hydrogens will diffuse through ATP synthase. ATP synthase is somewhat like a rotor. It's going to go around and around, and in going around, it is going to phosphorylate a DP with an inorganic phosphorus, making ATP. So the electron transport chain is the electrons flowing down the chain to the last oxygen electron acceptor. As they are going down the chain, we are creating a proton motive force in the intermembrane space. That is, again, the proton motive force. Chemiosmosis is the actual flowing of the protons, the hydrogen ions, through the ATP synthase and thus generating our ATP from the ADP plus our inorganic phosphorus. Notice that the electron transport chain does not in and of itself create ATP. It simply creates a proton motive force in the intermembrane space. As those diffuse through the ATP synthase, that's when we're generating our ATP, and that substage is called chemiosmosis. Now, here's the ATP synthase a little up close. Notice the intermembrane space. We've got many hydrogen ions or protons. They will flow through the ATP synthase, creating the movement of the rotor, and that is what will actually make ATP, and we are back inside the mitochondrial matrix. Now, the energy stored in the hydrogen gradient, the hydrogen ion gradient, is what is going to actually cause the ATP synthase. I want to show you this McGraw-Hill. It is a different interactive, and it just shows the whole cycle in a little different way.
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH. These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force. During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed. The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP. This enzyme allows protons to pass back into the cell and couples the energy released in this process to the phosphorylation of ADP to form ATP.
Okay, I should have mentioned that interactive is actually of a bacteria, so you may have noticed that the hydrogens were going outside the cell. Well, that's because bacteria don't have a mitochondrial, they don't have membrane-bound organelles. But in a eukaryote, the hydrogens would have gone to the intermembrane space of the mitochondria. But it was a good picture because it just showed things flowing just a little differently. Okay, we have just seen this. One thing I wanted to mention, during cellular respiration, aerobic cellular respiration, the energy generally flows in the following sequence. Glucose is oxidized. It reduces NAD plus to NADH. It is an electron carrier. It will carry the electrons and the hydrogen to the electron transport chain. The electron electron transport chain will create a proton motive force, which will turn our ATP, or which will turn our ATP synthase and thus generate our ATP. Now, it's very interesting that the cell, the mitochondria, is only 40% efficient in gleaning the potential energy from glucose. 60% is lost, it's wasted. You might think that's a high amount of waste, but the most efficient human-made machine is only 25% efficient. So where did the rest of the energy go? Well, most of the rest of the energy went off as heat. You are an endotherm. Your body remains warm. Well, the warmth is coming from the heat inefficiencies of the aerobic cellular respiration pathway. Okay, this is the last picture of this whole cycle. I wanted to mention something in your book. If you will turn to page 176, our new edition books have a different number of ATPs being generated. I don't know whether it's a misprint in our new edition. Last year's edition, this is a picture from last year's edition, and notice that glycolysis in your book reaps 2 ATP, the citric acid cycle reaps 2 ATP, but notice in your book it says that oxidative phosphorylation gleans 26 to 28 ATP, and altogether the process harvests 30 to 32 ATP. I believe that is a misprint. I went back and looked on the internet thinking, well, maybe there's been a scientific discovery since last edition, but I believe that's a misprint. So this is what I'm going to ask you to do. I'm going to ask you to take a pencil or a pen and very neatly cross through in your book under oxidative phosphorylation, where it says that you get about 26 to 28 ATP. You can cross through that and put 32 to 34 ATP, and then the final ATP, the maximum per glucose, should be 36 to 38. Now, I wanted just to mention some really interesting facts. Have you ever wondered what causes arsenic poisoning or cyanide poisoning? Well, arsenic poisoning actually interferes with one of the essential enzymes which catalyzes the oxidation of pyruvate to acetyl-CoA. When you stop the oxidation of pyruvate and into acetyl-CoA, you've essentially stopped aerobic cellular respiration, and of course, the brain dies. The cell dies, and therefore the person dies. Cyanide poisoning also inhibits an important enzyme, but it affects a cytochrome in the electron transport chain. And in so, it has the same effect. It stops the aerobic cellular respiration process. It stops the production of ATP, and so therefore the cell dies, and then the person ultimately dies because the brain cannot stay alive. Okay, we will pick up here next time I see you. Hopefully I'll be back from the Canadian Rockies, and I will be virtual. I'll be with you.